Uh, we have been discussing the role of epigenetics in contrast to the genetics of the human diseases. Now, today in this segment, we will discuss the DNA methylation in the tumor detection. That what is the role of methylation of DNA and how we can use it for detection of tumor in the human cells. Now, human tumors, they show the specific profile of hypermethylated tumor suppressor genes. The genes which have been suppressed by the hypermethylation of the DNA. And usually what happens that one or more genes are hypermethylated in every tumor type. There are different tumor types which we come across, and these are hypermethylated DNA, and we will discuss in this that how these genes they become the indicator of the tumors. For example, hypermethylated in gastrointestinal tumors, the genes like P16, INK4A, P14, ARF, MGMT, APC, and HMLH1, they are typical hypermethylated in gastrointestinal tumor, that is the colon, gastric, which they indicate if we detect this gene, then there is an indication of these tumors presence in the human. Now, aerodigestive tumor types are such as lungs, head and neck. They have the different pattern of hypermethylated genes. And these genes, they include DAPK, MGMT, P16INK4A, as discussed previously, but not HMLH1 or P14ARF. So if we don't detect these genes, then we get a specific pattern of hypermethylated genes present in the human tumor. Now, in the breast and ovarian cancer, BRCA1, GSTP1, and P16INK4A, previously, they are methylated in the breast and ovarian cancer. That is, if we find these genes, that is the BRCA1, GSTP1, and methylated in a sample, in a human tumor sample, we can indicate that this is the indication of breast and ovarian cancer presence in the human being. Now, DNA methylation markers, what are they? Now, these are the markers, they are usually indicators. And the use of DNA methylation markers in cancer diagnosis have some advantages over genetic marker. You see, because as I said that the epigenetic is a great advantageous tool for detection these cancer cells, these tumor cells in this. Now, there can be two approaches. One is in the diagnosis that we use the epigenetic, these methylated markers, or we can do with the genetic markers. That is, we find out the genetic markers present, and then we say that, there is a tumor present. Now, let's say that what is the system of the genetic marker? Now, that is in the genetic marker, what happens? Mutations. Mutation, they occur at the multiple sites. जो जीन है उसकी डिफरेंट साइट्स के ऊपर म्यूटेशन हो जाती है, डीएनए जो चेंज हो जाती है. While promoter hypermethylation occurs within the same region of the given gene in each form of cancer. ये एक फर्क जहन में बिठा लें कि क्या द म्यूटेशन अकर्स एट मल्टीपल साइट्स while the promoters they hypermethylation occurs within the same region of the given gene. एक जीन जो है उसके उसी हिस्से में वो जो है hypermethylation होगा and they form of a cancer cell. Second, the bisulfite modification of DNA जो है it enables the detection of DNA hypermethylation in a background of healthy cells and using usual amount of DNA while larger amount of DNA they are needed to detect the mutation. अब फ़र्क ये है कि हमें म्यूटेशन को डिटेक्ट करने के लिए लार्जर अमाउंट ऑफ डीएनए की ज़रूरत है. अगर बाइसल्फाइड मॉडिफिकेशन हो गई डीएनए की तो ये क्या करता है, इट अनेबल्स द डीएनए हाइपरमेथिलेशन. डीएनए हाइपरमेथिलेशन को ये मुहम्मेज करता है, उसकी तमीज़ करता है टू द बैकग्राउंड ऑफ ऑलरेडी हेल्दी सेल, जो पहले या जो सेहतमंद सेल्स हैं उनसे ये उनको डिफ्रेंशिएट करने में यूज़ होता है. Now CGP island hypermethylation is another, and this is used as a tool to detect cancer cells in all types of biopsies and the biological fluids. जो biological biopsies होती हैं टिश्यूज़ की, organs की या biological fluids की, to detect that whether there is a tumor present or not, then that is CGP island hypermethylation is detected, and they are usually, those biopsies are bronchoalveolar, lavage, lymph nodes, sputum, urine, semen, dental lavage, and saliva, blood, and stool. Not interesting biopsies, but they are used in the case to detect the tumors. Now, in the end of the last century, that in 1999, there has been a launch of new line of research for diagnosis of these cancer cells. And in 1999, what happened? That the first time screening was done for hypermethylated promoter loci in the serum of DNA from lung cancer patient. Lung cancer patient जो थे उनका serum लेके तो hypermethylated promoters loci they were detected to make the diagnosis of the tumor cells. Now another study, the septum 9 is another thing to study, and a single methylation marker is septin 9, and this is for colorectal cancer and could be detected with an high specificity. टेस्ट ऐसा होना चाहिए जिसमें स्पेसिफिसिटी, जिसमें रिजल्ट जो हैं वो बड़े क्लियर हों. चुनाचे इनमें 95% जो हैं वो क्लैरिटी वी कैन रीच इन द प्लाज्मा with sensitivity of 50 to 60% statistically in these cells. Now, another way is the gene silencing of the tumor suppressor genes. That is the early sign of malignancy. जब early sign malignancy का होगा तो उसमें क्या है कि gene silencing of tumor suppressor that can be done and often found in the early stage of tumor progression and is present in the premalignant lesion. जब premalignancy अभी जाहिर नहीं हुई तो उसमें ये है. Now, certain examples are, they include P16 INK4A, P14 ARF, APC, and MGMF. They are hypermethylation in the colorectal carcinomas and the aberrant hypermethylation of HML1 in a typical endomaterial or endomaterial hyperplasia. That is this is the gene which is used there. Now, DNA methylation, which is actually the epigenetic event, as I said earlier. Now, DNA methylation is the best known epigenetic marker in the pre-malignancy or premalignant event or precursor lesion. They could be very, very specific and useful for the early detection of cancer. So in this segment, we saw that how DNA methylation can be helpful in the early detection of the cancer, carcinomas, and the tumors in the human body.